All right. Good afternoon, everybody. I want to thank all of you for joining us here today, both in person and online. I'm really excited for this lecture. This is the second annual Klag Meoni lecture series. For those of you who don't know me, my name is Ashi Wiraratna. I'm the chair of biochemistry and molecular biology, and it's my pleasure to welcome you all to this special event. We're delighted and honored to have Dr. Melissa Davis, who is a world-renowned expert in health disparities, as our second speaker, I believe, in this distinguished series. So before we hear from Dr. Davis, I am going to share a little bit of background about this lecture. For those who are familiar and have been at the School of Public Health for some time, I see some of you in the audience. I know that you are very, very familiar with Mike Klag and Lucy Meoni, and they need no introduction. Mike was the former dean here at the School of Public Health. Many of you have had the privilege of working with both Mike and Lucy and calling them your friends. For newcomers to the School of Public Health, such as myself, let me highlight the remarkable impact that this couple has had on our institution. So Mike served as our dean for 12 years, from 2005 to 2017, and during his tenure, he guided the school through significant changes. He embraced technological advancements in education. He elevated our status as thought leaders and policy innovators. He expanded our faculty and fostered stronger connections both within Hopkins and with public health institutions across the country and indeed across the globe. Prior to his role as dean, Mike worked as a physician serving low-income patients in East Baltimore and conducted research on racial disparities in kidney disease. This background makes today's focus on health disparities particularly fitting, I believe. Lucy also has very deep roots at the Bloomberg School. She is an alumna with an SCM in biostatistics, and I see our current chair of biostatistics in the audience here. She initially joined Hopkins School of Medicine before she made the wiser choice and came over to a primary appointment at the School of Public Health. I'm probably not supposed to say that because I have appointments in both. Her contributions to the field are substantial, notably her leadership in the Precursor Study, which is an innovative longitudinal research project which began as an intriguing experiment and has yielded valuable data and significant discoveries. Both Lucy and Mike have been involved in this study and have collaborated on numerous publications and presentations. During Mike's deanship, Lucy played a crucial role both as a partner and an advocate for our school and helped to strengthen the relationships of Johns Hopkins Bloomberg School of Public Health with its supporters. It is worth mentioning that this lectureship was established through the collective efforts of our department chairs who wanted to create a meaningful tribute to say thank you to Mike and Lucy. Their initiative, along with contributions from faculty, staff, and friends, speaks volumes about the esteem in which you are both held, Mike and Lucy. This lecture series not only builds on your legacy, but it also reflects the profound impact you've had on our community. On behalf of everyone here, I thank you both for your ongoing dedication to our school and to the field of public health. Your contributions continue to shape and inspire our work. So can we give them all a round of applause, please? So normally I would introduce Dr. Davis, but as sort of a special treat, one of our new faculty at BMB was a former trainee of Melissa's, and so she will introduce her. But before I bring you up, Brittany, I'm going to ask Dr. Klag if you would like to come up and say a few words.
Well, Shani, they were wonderful comments. I wasn't prepared for that. I know Lucy and I are both touched. I just wanted to say thank you for you, Melissa, for coming and gracing us with your presence. We can't wait to hear what you have to say. But also to the donors who made this possible. And when I stepped down as dean, the chairs and the associate deans got together and came up with this idea of having a lectureship in our names. And I don't know whose idea it was to have it rotate among all the departments, but it was a great idea because if they'd asked me, then it would have been hard to choose one department. But BMB has always been close to my heart ever since I became dean, and you're the third chair that I've known of the department. And because it's one of the founding departments of our school, And one of the things that makes our school such a great school is the span of work that goes on here, from very basic, molecular mechanistic research up to policy. And I always felt that interaction of people working together, all united under that common mission of improving the health of people around the world, has been one of the things that's created the synergies that we have here. So I'm so happy that you came as dean. Eric, no, you're the chair. I was the dean, and so because it's hard to find somebody who can lead BMB effectively, because it has to be somebody who's a top-notch basic scientist, but can wrap their minds around the whole mission of the school. And so I think all of us are appreciative that you're here as leaders. So I think Lucy and I just wanted to say thank you, everybody who's here and listening, and especially to Melissa for giving the talk. So thank you.
Okay, good afternoon, everyone. So it is my great pleasure to introduce our distinguished speaker for today's inaugural Klag Meoni lecture, Dr. Melissa B. Davis. Dr. Davis is a trailblazer in the field of disparities genomics and is a leading expert in health inequities, making her an ideal choice for this prestigious lectureship. Dr. Davis currently serves as the director of the Institute of Translational Genomic Medicine and professor of microbiology, biochemistry, and immunology at Morehouse School of Medicine. She's also the inaugural Georgia Research Alliance Distinguished Professor at MSM, a testament to her exceptional contributions in cancer genomics research. Her academic journey began at Albany State University, where she earned her BS, followed by a PhD in molecular genetics from the University of Georgia. Go Dogs, I'm gonna have to say it. Dr. Davis's postdoctoral training at Yale School of Medicine and the University of Chicago laid the groundwork for her pioneering work in functional genomics, systems biology, and cancer health disparities. Dr. Davis's research focuses on uncovering the complex interplay between genetic ancestry, tumor biology, and health outcomes, particularly in breast cancer among Black women. Her groundbreaking findings have established a new paradigm for studying biological factors in cancer outcomes as they relate to genetic ancestry. She has discovered crucial links between African ancestry and tumor burdens that disproportionately affect people across the African diaspora. As the scientific director of the International Center for the Study of Breast Cancer Subtypes, you'd think I would have that by now, and the director of health equity for the Englander Institute of Precision Medicine at Weill Cornell, Dr. Davis has been instrumental in advancing our understanding of cancer genomics in diverse populations. Her work is pivotal in addressing the stark disparity that Black women in the United States experience, as they are 41% more likely to die from breast cancer than white women. Dr. Davis's research extends beyond just breast cancer, encompassing prostate and gynecological cancers. She employs an innovative approach, utilizing quantified ancestry to disentangle genetic and environmental influences on tumor biology among different racial and ethnic groups. This includes investigating epigenetic cell signaling and immunological responses in the tumor environment. Her commitment to increasing diversity in genomics research is evident in her leadership of the Polyethnic 1000 Project as an ethnicity scholar for the New York Genome Center. This initiative aims to expand our knowledge of genomic profiles in underrepresented minority cancer patients, ultimately working toward more equitable therapeutic and diagnostic options. Dr. Davis's work exemplifies how the inclusion of diverse ethnic groups in research can lead to the discovery of novel genetic risk factors and gene network modifications, resulting in a deeper understanding of unique tumor biology. For this work, she has been awarded a CRUK Grand Cancer Challenge Grant. Let's just give her a round of applause for that. This is a wide-scale collaborative grant that brings together multiple PIs from all over the world, including collaborators here at Johns Hopkins, to tackle this problem. So in addition to her being an amazing person and researcher, she has been a mentor for me personally for many, many years, I would absolutely not be standing here today without her. So please join me in welcoming a true pioneer in health equity research, a dedicated scientist working tirelessly to bridge the gap in cancer outcomes, and an inspiring leader in the field of genomics, Dr. Melissa B. Davis.
Wow, thank you so much for that introduction, Brittany, my lab kid, my first lab kid. And I'm very proud of her. So today, actually, she gave pretty much a summary of my talk. So I'm just going to demonstrate everything she just told you about. So the title of my talk includes sort of what I call the dark side, because the gene we study, one of them is the DARC gene. But I'm also a Star Wars fan, so I'm a sci-fi fanatic. One of the things that I hope that as you depart from this lecture today is a deeper appreciation of the fact that we are trying to utilize wherever we can find advances in technology to help improve the outcomes of populations that we described in many different ways: marginalized, underserved, underrepresented, but that in some aspects along the way, it's been quite challenging to do this work. So I'm going to tell you what we've done, but I'm also going to give you a little background about how we've overcome some of the limitations through the work that we've been doing and through the partnerships that we've actually been able to forge along the way. So disparities, why we care, how you care matters, and how we're cared for matters. So this is the type of research that is forthcoming, that of course, there are socioeconomic barriers that impede whether or not patients survive an outcome or survive a diagnosis. Deeply embedded in the healthcare system is a reflection of the world that we live in. So racial segregation across the United States had many different consequences on not only the socioeconomic status of populations that were marginalized by segregation, but also the healthcare system was also marginalized through those segregation practices. What we know is that Black patients in the US were more likely to be located in areas where the clinics have serious deficiencies, reduced staffing, greater financial vulnerability. In some cases, they didn't even have necessarily the state-of-the-art technology to deploy standard of care. What I'm showing you here is a map of the United States, county by county, showing poverty rates. And now what I've overlaid here is where the majority of the Black population lives in the United States. And what you can definitely appreciate is that around the Southeast, where this Black belt, which was originally called the Black belt because of the soil and the richness of the soil in the land, but now has become a socio-political term referencing where the Black population lives in the United States, overlaps with this band of poverty in that same region. Some of this has a lot to do with rurality, but the persistent segregation certainly does lead to some of the disparities that we see. However, when we take a closer look at how desegregation, also now in the healthcare system, has improved access to socioeconomic status and higher strata in this marginalized population, we still see persistent mortality disparities. So what I'm showing you here now on the left is the breakdown of mortality rates in prostate and breast cancer in the United States, and the blue lines represent the Black population. The lighter color represents the Black population that lives in affluent counties. So these are patients who are at the highest SES strata. So in those counties, we don't have the same barriers of access to technology and care, but we still see that despite that, the Black patients who live in affluence still have a higher mortality than the white patients who live in poverty. And so that means this is not necessarily a poverty issue, but there's some persistent factor that continues to drive this disparity. Now, interestingly, when we look at the breast cancer line, and I don't know if the pointer will show up, but that's okay. Around 1990 is when the disparity emerged. Prior to that, it didn't matter if you were Black or white, you had pretty much the same opportunity to survive a breast cancer diagnosis. At that same time, what we see is the advent of standard of care where endocrine targeted therapies were introduced in the cancer setting for breast cancer. That created this stark decline, specifically in the white population with lower mortality rates. But for the next decade or two, we see this persistently higher mortality in the Black population that we don't actually see it to start to decline until the early 2000s. So that delay in the advent of technology, of thinking about targeted therapy, of even defining what different types of breast cancer are, is what feeds into, still today, the disparity that's consistently seen in prostate and breast cancer. Now, if we think about all of the different facets of drivers of just general health, all of this also feeds into whether or not a patient is going to overcome and survive a breast cancer diagnosis. As you can see from the wheel, it spans from behavioral, environmental exposures. Well, we know carcinogens certainly have overexposures in certain areas, and I'm going to get into that a little bit. But what we're starting to have a better appreciation of is the interplay between the biological and the psychosocial. But when we look at a mediating factor analysis, we find that tumor characteristics actually plays a very significant influential role in racial disparities, about 24%. Now, one of those tumor characteristics is hormone receptor status. We know that triple negative breast cancer, which is one of the most aggressive forms of breast cancer, is more likely to be diagnosed in Black women. But we also know that even in the ER positive category, we see more aggressive forms of the disease, even refractory tumors, so they don't necessarily respond well to treatment. Now, we focused primarily on triple negative breast cancer early on, and as you can see here, Dr. Clayton Yates was one of my first collaborators way back at UGA. Brittany will remember the time no one had any money, so we just pooled all our money together, had group lab meetings, and we tried our best to overcome the barriers to doing the research at all. Now, one of the things we observed in our communities was, again, this higher incidence of triple negative breast cancer, this earlier onset in African American women. And in a complex disease like cancer, earlier onset tends to hint at there being a predisposition for this type of phenotype or disease. So what we wanted to understand was, if we looked across the population and did this comparison between white and African Americans with triple negative breast cancer specifically, what might be some differences? One of the first differences we saw when we reanalyzed TCGA data, so that's the Cancer Genome Atlas, we found that African Americans with triple negative breast cancer were also more likely to have a fourth hormone receptor negativity androgen receptor. So we coined it quadruple negative breast cancer. Quadruple negative breast cancer also has an even earlier onset than triple negative breast cancer, again, leaning towards there being a genetic predisposition. But then with every bioinformatics tool that we could use to define the subtypes of cancer, the Black women had a more aggressive form. So there must be something different in the biology at a population level. And this is something that had never really been discussed before. So then we, again, pooled our resources together and started looking at independent cohorts, at cell lines, and setting up experiments that allowed us to look at the cellular pathways and the mechanisms that might be different between quadruple and triple negative breast cancer. In these early papers, we found things like the P10, the EGFR pathway. We also looked at epigenetic mechanisms such as microRNAs, and we assessed at a population level that there was a pattern of microRNA expression, a cluster, if you will, of them that were uniquely expressed in African American women. Looking closer at this cluster, one of the core microRNAs happened to be in the canonical MYC pathway. MYC, as we know, is an oncogene, but then when we looked at the clinical outcome of whether or not patients expressed this particular microRNA, it was only clinically relevant in African American women. So all of this data cumulatively tells us that there are underlying differences in the tumor biology between these populations of women that we haven't really captured in a mechanistic, systematic way to help inform some of the targeted therapies that were currently being utilized. And why is that important? Well, first of all, we decided to take a global approach, something called oncologic anthropology, which was a term coined by my previous research partner. We find that some of the highest mortality burdens exist in Africa. Some of that is obvious. because of lifespan and other challenges on the continent. But specifically for triple negative breast cancer, we see that the highest rate or incidence of triple negative breast cancer globally is in sub-Saharan Africa. Then in every country where we can separate the population by self-reported ancestry or the Black population from the white population, it's the Black portion of the population that has the highest rate of triple negative breast cancer. And then we actually measured ancestry, because all of this, as my PhD advisor at the time would say, is just hand waving and conjecture. It's just correlated. It's a coincidence. So we worked with Dr. Rick Kittles and we measured West African ancestry in our multi-ethnic cohort, which included white Americans, Black Americans, East and West Africans. And what we found was that the portion of the population that had the highest amount of West African ancestry was also more likely to have triple negative breast cancer than any other type of breast cancer. So that highlighted for the first time that the genetic background of patients of West African ancestry may harbor some specific predisposition to this specific biology of breast tumors. And why might that be? When we think about the African diaspora, it's unlike any other human population on the planet. And that is because of the unique gene flow that has occurred due to the transatlantic slave trade. Now, when we think about other races and the migration pattern of humans out of Africa, for instance, or even other founder populations where a single event, where migration of a subgroup of the population simply moves from one location to another, and then subsequent generations create typical gene flow for each subsequent generation. Instead, for the African diaspora, what we see is that over hundreds of years, instead of just a small population, millions of Africans were displaced. So if over 13 million Africans were displaced over generations, over centuries, then what we see here is a unique flow of that genetic background, which is why when we separate each population by ancestry, we can see that in that particular group, we have this allele that is yet unknown, but in the background of this ancestral lineage. And what does that mean? What does it mean that having a certain ancestry in your admixed genome harbors a likelihood of having a certain disease? We know that if you're of an admixed, and there is no such thing as a non-admixed population, honestly, this day and age. But your ancestry is stochastically cast across all of your chromosomes. We can't really say for sure just by looking at you what percentage of African ancestry you have, or even where your African ancestry might reside on which chromosome. So what we do is employ something we call applied haplotyping, where localized ancestry measures allows us to identify loci that are enriched for that genetic background. So in similar ways of the transmission experiments that Marie Claire King used to identify the BRCA gene, this allows us to identify loci in the genome that are shared ancestral or genetic background across the population of patients who have triple negative breast cancer, for instance. The way we apply this information is really important, because what you'll see currently as more studies start to appreciate genetic ancestry and measure ancestry is that they utilize sort of an arbitrary cutoff of percentages, right? So if you have only 10%, then you're not in the African category. But we don't necessarily want to redraw boxes to re-identify who people are. Instead, even the one who only has 10% still has that genetic background at the appropriate locus for us to identify where the loci is. So in this way, we are actually increasing our power to identify the region of interest, that is what we're looking for, by not casting people out of the study just because they don't have a high enough ancestry from Africa, for instance. But why does this all matter? So if we look at triple negative breast cancer and we compare white and African American women, and this is about 20 years worth of data spanning across Detroit through Henry Ford Health Systems and the New York City area and Newark Presbyterian hospitals. What we wanted to understand was whether screening made a difference. If we could find an earlier detection method, could this save the African American population? Is it because of low screening rates? We actually know from health services data, that African Americans are screened at the same rate, if not even more so, as a population as a whole. Some of that gets a little bit in the weeds of the quality of the screening, depending on where you are. But for our cohort, these women were all in the same hospital system, being seen by the same radiologists, the same oncologists, same surgeons. And so we did not see a huge difference in whether or not the triple negative breast cancer cases were screen detected or non-screen detected. Now, what we know about screen detection is that if you do have a diagnosis that comes from screening, it's usually going to be really early. You don't know that there's anything wrong. The screening, the mammography is what actually highlighted the mass. For non-screen detected cancers, these are usually later stage cancers because something is awry. The patient is compelled to come into the clinic because they have identified something. And so then that's usually a later stage. So then across the whole population, we expect to see a lower survival rate in the non-screen detected category. Breaking this across race groups, even in the white population, we see a lower survival in non-screen detected cancers. What we did not expect to see was this huge gap in survival in the Black women who had non-screen detected cancer. Now, clinicians see this and they think, oh, we got to screen even more because we need all the Black patients on the blue line. But for me, I see this and I think the treatment is not working the same in this population of women. And maybe it has something to do with all of those findings that we've recently seen where there are differences in the biology. But some of this even has to do with overall survival, and given the cardiotoxicity that comes with the systemic chemotherapy that has been standard of care for triple negative breast cancer for decades. We know from pharmacogenetics and pharmacogenomics that there are alleles and pathways that are a risk to cardiotoxicity for certain types of chemotherapy, such as anthracyclines. And when we look at the allele frequency across the global population, and this comes from 1,000 Genomes data, the highest frequency of these alleles are in people of African descent, African American, African, Dominican. Even on the right, the far right, we see even in South Asian, Latin American women. So these are non-European ancestry groups. However, we also know that these women or this population, these populations are likely to not be in the clinical trial. So we don't see that incompatibility at the clinical trial level. We only see it after it's deployed as a standard of care, and then it translates in the population as a lower survival in these particular populations. But when it's confounded with information like barriers to access, issues with the tumor biology not being responsive, it's very difficult to disentangle what is actually happening. Identifying genetic factors for cancer risk should be an easy task at this day and age. Now, early on, Marie Claire King and others had to use pedigree analyses, and what a phenomenal job. But in order to use that type of analysis, you have to have a large family with known relatedness across multiple generations and very well curated health history. And our social history in the United States will preclude any of those things from being feasible. But GWAS studies should have saved us because in those, we don't need people to be related. We just need a large group of people who have the phenotype and who don't. And then we identify which genotypes are associated with the phenotype or the disease. But unfortunately, we have a bias in sampling, convenience sampling. So the people who walk into the doors of the academic centers that are doing the GWAS studies, and unfortunately, what this leads to is a lack of information and statistical selection bias. And it leads to inaccurate clinical tools. So here's a snapshot of the data that currently exists right now in our public domain and of genomic data. And what you see here is that the European group is far more represented by 90% or more. And this, said a different way, is that less than 10% of the data represents 80% of the world, because the European population is not the global majority. And so we've actually built our entire knowledge base on genetic predisposition and genetic risk on a single population that is heterogeneous in itself, actually, and completely ignored the larger group of the global world. And so what that represents then in clinic is, for instance, on the right, what I'm showing you is a higher rate of variance of unknown significance. So variant of unknown significance is unknown because it hasn't been seen before. And so if it hasn't been seen before, then we can't have any action. Unknown significance also means unactionable. And so if you're of non-European ancestry, you're more likely to have one of these variants. That means that the patients have a mutation in a known cancer gene, but it's a mutation we've never seen before and we can't do anything about it. Whereas if it were a known pathogenic allele, because we've had better representation in GWAS studies, then we'd be able to open up an entire arm of the healthcare system to increase screening in the population that is at risk, but we haven't yet really fully defined what being at risk is. So in steps Team Standby. So this is the name of our Cancer Grand Challenge team, and it stands for Societal Ancestry and Molecular Biology Analyses of Inequities. It's made up of 5 different work packages, starting with social and societal determinants, exposomics, then genomics, then tumor immunology, and then patient involvement and dissemination. I'm going to walk you through not only what each one of our work packages is doing, but some of the data that we had generated, which was the foundation of the preliminary and previous studies that led to the team coming together. So our logo is a lens, and it's focused on Africa. And each part of the aperture represents the methodology or the discipline that we're using in the team. We focus on breast, pancreatic, and prostate cancer, again, because the global burden for these diseases are in this diaspora. And then when we think about how we've done comparative studies in the past, we've been able to uncover that there are genes that are related to ancestry. It could be African, it could be European, and I would even say if we look in any other population, we're going to find ancestral informative markers that are also EQTLs. And so if you deploy this type of method, you're going to find differences related to ancestry. But here at the bottom, this blue circle represents genes that we also found was strongly associated with being African American, but not associated with being of African descent. And when we look at, because in this study, we actually compared African Americans to West and East Africans. Now, when we look at what those race-specific gene networks are, what we found was that these represented obesity, diabetes, heart disease, other metabolic disorders, insulin insensitivities, and these were in the tumors of these women. So that means that the comorbidities that are systemically plaguing a population of people, because I'm sure you've heard the statistics that obesity, diabetes, hypertension, all of these are more prevalent in the Black population. This means that these pathways are actually present in the tumor space. But again, they've never been a part of any clinical trial because systematically you're ineligible to be in a clinical trial if you're obese, if you have high diabetes, if you have high blood pressure. And so then the tumor biology that underlays these patients, and we're talking about hundreds of genes, have never been targeted in therapy, but certainly haven't even been accounted for. And so what the first work package team leads will focus on is expanding their research. So this is Dr. Lauren McCullough and Tiffany Carson, and they are the co-leads for work package 1. They'll be helping us focus on the unique aspects of all of the regions where we plan on doing prospective recruitment. This would include tribal regions in Ghana, Nigeria, and Ethiopia, as well as the Black British population, and of course, the non-Hispanic African-American population in the US. Now, our approach is to have from an area level all the way down to an individualized level, a new framing of how social determinants is measured and what are the actual factors that are relevant in this population. Because again, up to this point, in social determinants work, it's always been centralized around being in a deprived environment, that the social determinants that are impacting the Black population are hand in hand with them being marginalized into an area that is deprived or overexposed to physical environmental exposures. But what Lauren will tell you is that Black women at the highest SES strata have 126% more likelihood of dying from a breast cancer diagnosis. So that means living in that affluent county does not necessarily reflect, for instance, census tract data that is currently usually the canonical way social deprivation is defined. So instead, what we'll be looking at is societal trauma. So not just whether or not you live in a redlined area, but were you impacted by a natural disaster that displaced your family? Are you suffering from social isolation? Do you have other aspects, especially in Africa, that threaten your life on a daily basis. And so now you have a different form of stress. It's not related to discrimination per se or racial discrimination, but it's related to surviving, a harsh and hostile environment. And then in the highest SES strata, the other aspects of psychosocial health that Black professionals have to deal with, microaggressions, racial profiling, the implicit biases in their employment and in the healthcare space. How can we measure those aspects of their lived experience and quantify them to now have this be an explicit part of their risk analysis? Of course, structural racism is real. We know that redlining effects last decades after. Looking at life expectancy in Manhattan, for instance, in a 30-block radius, is a 50-year lifespan difference. And why would that be? What this map represents are the areas where we know there are hazardous waste or health threats to the people who live in these areas. So the green and the yellow and the orange and the numbers inside represent where health toxic areas are in these communities. And what we see is that they are overlaid where we know marginalized or redlined areas are. So what is the exposure in these areas? That brings us to our work package 2. When we think about where people live, whether they have any control over it or not, so for instance, lifestyle exposures due to smoking, alcohol use, or beauty care product use. We know that there are signatures of their exposure that remain in their body for some time, and that this can change physiological pathways. So what Dr. Miller and Dr. Meoni will be working on is using forms of mass spectrometry to measure in circulating plasma what some of these particulates and molecules are, the compounds of interest, he calls them, to identify what is circulating in our cohort. So of the 40,000 people who are enrolled, we'll be measuring using mass spectrometry over 200,000 different data points to understand what in their environment is seeping into their blood. In previous studies, Dr. Miller was able to, using a cohort in New York, he's at Columbia University, in an Alzheimer's study. He was able to uncover particulates that were unique to different ethnic groups. Now, geographically, they're also unique to certain neighborhoods. And so what he found was that a population of patients who lived in the Manhattan area lived very close in proximity to a bus depot. And so the air pollution from the bus depot was imprinted, I guess you could say, or just present in higher amounts in the patients for PM 2.5. Now, thinking about air pollution, we know that some of the air pollution that patients are exposed to in New York won't necessarily be the same source as the patients who are exposed to air pollution in Ghana or Nigeria, but down to the particulate level, PM 2.5 might be the thing in the air pollutant, no matter where it's from, whether it's coal mines, whether it's car exhaust, that we may be able to discover across the entire global diaspora. And that's the strength of having a global cohort. This is work package 3. I'm sorry, I was looking at Nicholas's picture is jumbled there. Don't tell him. Oh, it's right here. Okay, great. So in our genomics work package, we'll be focused on a lot of stuff. So this is my area. And what we want to do is, first of all, create a cohort of data and data integration that's never existed before on this population. So we'll be doing whole genome sequencing on their germline. We'll also be doing whole genome sequencing on tumors. We plan to do long read technology. We have Oxford Nanopore Technologies right now on the abstract, but we might switch to a different technique. But the point is to be able to measure at a long range structural determinants or structural variation in the genome, complex rearrangements, for instance, but also epigenetic signatures such as DNA methylation that are related to everything that we found in work package 1 and work package 2. And I'm going to show you some of the data coming up here in a moment. But one of the other exciting aspects of Work Package 1 is the fact that we'll be building our own human genome references that represent the regions of Africa that we are recruiting from, but that do not have an appropriate genome reference with which to align all of our data. And I'm going to show you some key aspects to that here in a moment. So one of the things that we discovered, again, working with Dr. Yates, is when we did comparative analyses, initially we could only use self-identified race to separate the population. And even using self-reported race, you can clearly see that there's a difference in gene expression, but that difference becomes much more clear when we strip away race and use genetic ancestry. This is because these are genetic components that are driving the expression. The other things that are driving differences in expression might not be genetic germline variations, but could be because of variations in the population. But before we can disentangle what is environmental and what is genetic background, we need to fill the gap. We don't have enough data to actually do this with a rigorous statistical analysis. So therein steps the New York Genome Center, starting what we call the Polyethnic 1000 Project. Now, the Polyethnic 1000 Project was meant to allow us to have a public data set of 1,000 patients of cancer who the only inclusion criteria was that they did not identify as white or European. Now, our cancer discovery paper came out of the Polyethnic 1000 findings, and I like to joke and say we actually made the cover of Cancer Discovery. So not only was this the top cancer journal to publish in, but our work was featured on the cover. So if you get a gift from me, it'll be wrapped in the cover of Cancer Discovery. And I did get permission from the editor. I just can't sell it, but I can use it. So, well, this is figure number 1 in the paper, and it's one of my favorite figures because one of the things we discovered along the way of simply measuring genetic ancestry was, first of all, reifying the fact that African Americans have a broad range of admixture of European ancestry. But then in the bottom panel, what we're showing you is the regional ancestry, not just the global East Asian, South Asian, African, but regions of Africa, regions of South Asia, so on and so forth. And what we found was that for African Americans, the origin of their European ancestry was so diverse. And in each individual, every region of Africa was represented in their African origin. And again, that is that's our social history here in the United States, again, from the slave trade. What we also found was that our Ethiopian population had about 40% European admixture. So while Ethiopia touts to be the only African nation that wasn't colonized, they did have a unique social history with Italians in Eritrea. And that source of their European ancestry is full on just about all 100% Italian ancestry. And then in our European cohort, we do see, again, the source and origin of European ancestry vary. And so the point to this is that the genetic background is very important, and it's something that we've been ignoring full out this whole time. But now we have tools to not only measure it, but utilize it in our tools. So when we identified, for instance, a subset of genes that were both regulated by whether the region was of African or European descent, so that overlap in the middle, the expression pattern was the complete dichotomous opposite for that set of genes. That means that there's an EQTL that maybe arose in Africa, or maybe it arose in the European lineage after Africa, but it clearly regulates a subset of genes very distinctly. And now we want to identify what those alleles are, what those ancestral informative markers are. As we look at mutational signatures in the somatic tissue of our tumors, we're also finding that ancestry is associated with specific mutational signatures. So what I'm showing here on the left, if you can see at the top, which is the high African group, and the bottom is the low African group, the blue box represents mutation signature 1, which is a signature of aging. But this population is much younger, so that means that they have an aging imprint on their tumor that coincides with what we already know as premature aging in this population, again, associated with weathering and allostatic load and things like that. And it's been shown genetically through other types of assays, such as telomere shortening, so on and so forth. On the right, what I'm showing you is a really cool finding that's still emerging for an African-specific allele of a gene that is involved in DNA repair. And there are two signatures that are starting to associate with the African version of the gene, which is the T allele at the top, which is signature 5 and signature 16. Now, ANCL1 is actually a LIM3 ortholog, and its normal function is the resolution of chromatin bridges. And so in the absence of, or maybe the overactivity of this LIM3 gene, what we anticipate is that potentially there might be a global genetic instability, but certainly a higher likelihood of having chromatin bridges, specifically in tumors. And so this destabilization could potentially, present itself as a unique mutational signature. Now, when we identified ANCL1 as a possible DNA repair gene, it was interesting because previous studies had shown that the gene had an allele that was associated with protection against triple-negative breast cancer. But that was the European version of the gene. And so the gene had been largely ignored because every GWAS study showed that this ANCL1 allele was a protective allele. But the alternate allele, which is in higher frequency in ancestries of African descent, is a risk allele. And so that's what this scientific reports paper shows. We actually modeled the mutation and showed the structural differences between the African versus the European allele. And interestingly, we also showed that if we knew ancestry and we knew that the gene was highly expressed, that it even showed a survival difference. So that tells us that the African version of the gene is doing something different than the European version of the gene. And we wonder then if that's what we're starting to see when we see this distinction in the mutation signatures from having the African versus the European version of this DNA repair gene. In fact, we wonder if the cluster that defines signature 5 and signature 16 was, well defined in the AI model. But that's another story for another day. But we do know that these two signatures cluster together in the original algorithm that defined it. But we did not necessarily see that signature typically in breast cancer, but instead in other types of cancers such as stomach, bladder, pancreas, and lung, which are typically associated with external exposures. All right. The next story I want to tell you is around the Center for the Study of Non-Coding Cancer Genomes. Now, this was a study that was funded again through the New York Genome Center, and it was a pet project of mine up until we actually got funding. So this is a lot of information, but follow along. So in 2019, there was a paper in Nature Genetics that, through the CAPA consortium, which was asthma among African ancestry populations, revealed that if they did whole genome sequencing, that they could identify regions of the genome that were not present in the human genome reference. Okay, so there were contigs, over 125,000 new contigs of genome sequences that have never been in the human reference. That means we've never interrogated them in any of the sequencing platforms that we've ever done. So what we did was, first of all, ask the question, well, if we take some of our unmapped reads, which is shown here in the upper right quadrant, which is typical in any sequencing alignment, there's always going to be a bucket of reads that just doesn't align to the human reference genome. And we put them over to the side and we call them unmapped. And depending on the source of your tissue, it might be microbial genes, it might be chicken from lunch, if it's saliva, who knows? But we just put it off to the side because it didn't match anything in the human genome, and so it's irrelevant. But we took those unmapped reads and we asked the question, well, do they map to these new contigs? And almost a third of them did. And so that tells us that the express sequences that we've been ignoring over all of these different studies are actually genes in the genome that we've never interrogated in any study before. But even more exciting, we took those same contigs and asked the question, are there CPG islands in these new regions of the genome that have never been interrogated? And the answer is yes. We found 8,000 potential new CPG islands across 6,300 contigs that are these new contigs. And then when we overlap the contigs with the expression with the contigs with the CPG islands, we found about 1,100 new regions of the genome that have not only expressed sequence, but also have epigenetic modifying potential. And so I'm excited about this because we'll be digging a little deeper in whether or not they're functionally relevant. And some of that information is also going to help explain why, for instance, polygenic risk scores aren't the same across populations, because there are regions of the genome that we haven't included in our modifying analyses. In the fourth generating work package, you'll see a familiar face here. Dr. Clayton Yates is one of the co-leads. By the time we get to this work package, we will have amassed a huge amount of information about the patients, both at the area and individual level of their social and societal health, at their exposure level from the exposome, and now from the genome expression and whole genome sequencing. And now in this work package, what we hope to do is integrate all of that information to help us understand exactly how the tumors are different. So not only the cellularity and whether or not there is an immune signature, but the state of all of those different cells. And then how, for instance, any of those factors that we've established in the first 3 work packages may impact this tumor biology. Now, some of the precedent that we've already established was using bulk RNA-seq. This was a different paper than the one that I just showed you. This was part of the cancer discovery paper. So using deconvolution in that paper, we showed that people of African ancestry had a stronger immune response. They had a higher count of tumor-associated leukocytes. Brittany may remember the painful multiple IHC methods here. This is before there was a Koya and like multiplex immunological methods. She had to actually do this by hand. Thank you. We showed in the independent cohort that even by cell markers, we saw this stronger infiltrate of immune cells in our West African, specifically West African and African American patients. But this is counterintuitive because we know from previous clinical studies, where the broad population is considered, having more tumor-associated leukocytes is supposed to have a better survival opportunity. But what we see is that in African Americans versus European Americans, African Americans completely lose the survival advantage of having this higher immune infiltrate. We believe this is because, and this is where the dark side comes in, we believe this is because there's a unique tumor immune response due to something that is embedded in West African ancestry. And so now we're using fancier tools to define what that tumor immunological phenotype is. Now, DARC is a gene that is also the founding, the underlying gene of a blood group called Duffy-Null. Now, Duffy-Null is a mutation that arose in sub-Saharan West Africa. It expanded across Africa to the East African shore through the Bantu expansion. But it was fixed at 100% frequency in that population. And then when we look across the globe, we see this higher rate, again, that tracks with the displacement of Africans during the transatlantic slave trade. So now where we see people of African descent, we see that there's a higher frequency of this particular allele. Now, the mutation actually only removes the protein, which is also called ACKR1, but it's not as fun to use that name. But it removes the expression off of red blood cells. Now, normally on those red blood cells, the protein would act as a chemokine sink. It would actually remove systemic inflammatory chemokines from circulation. And this is what's supposed to happen. If you have an inflammatory event, it should be a spike that goes down almost immediately, or at least within a certain amount of time. If you have the Duffy-Null mutation, what you also have is chronic systemic inflammation because you don't have the ability to bring levels back down because the protein is missing. Now, it was fixed in the population because it also confers an immunity to malaria, at least through P-Vivax. What we also know, though, however, is that the red blood cells aren't the only place DARC is expressed. It's also expressed on endothelial cells, on blood vessels, specifically capillary venules, which is where immune cells tend to exit the bloodstream and enter into interstitial space. We actually know that this is because of co-binding of some of those chemokines. So now when we look at the expression of DARC, for instance, in the cancer space, what we see is that the expression of DARC at a gene dose dependent manner corresponds with higher immune cell infiltrates, probably from that endothelial expression. But we also found that there was a higher amount of inflammation in tumors that had a high level of DARC expression. So now as we look at the composition of tumors and identify whether or not either the patient is Duffy null or Duffy positive or the tumor itself has a high expression of DARC, we're finding unique immune cell types that are more prevalent in a DARC negative versus a DARC positive environment. And looking to the future, our strategy will be to integrate all of this data. So the beauty of SAMBI is the interconnectedness of it all. For instance, if I were to describe what my dream output deliverables would be, it would be epigenetics of exposomics. Molecular exposome matched to social determinants of health. This will be the first time we can actually quantify with a metric that is a biomarker how social determinants impact tumor biology, for instance. This is displayed beautifully in one of our studies where we are looking at the geospatial to tumor spatial imprint of how, for instance, food insecurity, environmental exposure, stress, and societal factors directly impacts not only the comorbid status of the metabolic disorders, but also impacts these particular pathways. So I'm really excited about this because structural racism has often been done in a silo away from biology. And we often hear this sort of conjecture of either or, which one is more, and they do not exist in mutual exclusion. This is the current state of our map in Detroit and New York City. And as you can see, we have whole genome data that is overlaid with where patients may have been living in redlined areas in these regions. I can't show you the data yet. Haven't gotten a thumbs up yet to tell you, but we do know that there are mutational signatures that are clustered around specific regions in redlined areas that are directly related to whether or not they are overexposed to certain health threats. And so these spikes of disparate incidents that you see in the larger circles may finally be defined. And if we can define these, I believe what we'll be able to ultimately show is that biomarker analysis that can predict how African ancestry, so Duffy null status more specifically, can help us identify chemokine signatures that we all know that actually deactivate immune cells, are again linked back to dietary indices that we've also shown in West African women. So this is the evidence how structural racism can modify tumor biology more directly. But if we deconvolve all of that information, what we're also saying is this is a blueprint for precision prevention. Because if I can tell you that the diet that you're eating, and we already try to do this, if I can tell you that the diet that you're eating can be monitored at a biomarker level as a cancer patient, then in this specific population, we'll have the evidence necessary to be able to train, if you will, immune cells to no longer be deactivated, but have the type of immunological response that these patients need. In order to do that, we have to do more than patient engagement. This is my last work package, and this is where I end. But patient recruitment is essential to equity research. But we're going to do more than just recruitment. This work package is actually led by patient survivors. So Ricky Fairley is a metastatic triple negative breast cancer survivor. And she's leading a team of survivors across prostate, breast, and pancreatic cancer. We gave them a budget to make sure that, first of all, we flip the power structure. So they'll go in front of us in the community and they'll ask the patients what their concerns are, particularly where environmental exposures are concerned. And then we'll educate them about all of the things I just described to you. So what is mass spectrometry? What is epigenetics? Why do we care about societal trauma? What do all those things really mean to them? And that'll help us refine all the tools that we're building and certainly help us to interpret at a much relevant level what we're actually seeing. One of the papers that they're working on right now is the community's response to the NASM report. How do they feel about whether or not we say they're of a certain genetic ancestry versus genetic similarity? Feels an awful lot like someone redefining who I am, and maybe that's not exactly what needs to happen. And so for policy advocacy for equity in cancer care to work, we have to empower patient voices. And a lot of what Work Package 5 will be doing is just that. So every scientific paper we publish, they'll publish a companion paper for the community. And we hope that by that sort of bidirectional information gathering and flow of knowledge, we'll finally overcome some of the trivial aspects of disparities, but certainly then empower our communities to be more active in overcoming the disparities that leads to less precision in precision medicine, but certainly not even fully understanding how in the diverse global population, all of these factors can be different depending on your genetic background, depending on your identity, depending on where you live, and depending on how you're treated and where you're treated. I'm going to end on this. So this is the capacity building that we're doing in lower and middle income countries. This represents one of the hospital sites where Brittany actually came with me. One of these pictures might represent one of the things we did there, but Kofanochi Teaching Hospital in Kumasi, Ghana is where my second lab exists. And that's one of my happy places. And with that, I'd like to acknowledge all of our funders and our collaborators, and hopefully I've left a little time for some questions. Thank you.
And Becky, are you going to take questions from Zoom at the end?
I mean, that was so incredible. So thanks so much for sharing all of that. It's
Thank
really
you.
inspiring. I'm curious, one of the challenges with cancer treatment is, and of course you know, is trying to get the representation in clinical trials
Mhm.
so that we know what, is going to be approved, is going to help the general population. What strategies do you think can overcome that hurdle to finally get the representation where it should be?
So Work Package 5 is one of our strategies. So Ricky has started her own clinical trial study called Why We Trial. And over the last 2 years, she has enrolled over 19,000 African Americans into her cancer trial registry. Amazing, right? And it's because it's led by patients who are fighting misinformation. Like some of this is trust, mistrust, well-earned mistrust, and some of this is misinformation, and some of it is simply that the areas where people are most treated, if you will, don't have clinical trials. And so her group helps to navigate them to places that they can be a part of a clinical trial. And what I think is amazing is she has this team of survivor nurses who also man a 24-hour hotline for anyone who has questions about clinical trials or their treatment. So when the doctor's office tells you to go through your proper channels, they call Ricky's line, and there's an oncology nurse there to answer the questions. So working with the community through patient advocates, I think, is going to be the key.
That's amazing. Thank you.
Thank you. Yeah, that was a great talk, and you crammed so much information. You don't look old enough to have done all this. I just was wondering.
I started when I was 5.
The DARC, the dull, the Duffy null phenotype. So African Americans are at higher risk for a whole variety of diseases, especially like kidney disease and some forms of cardiovascular disease.
Yeah.
So it's reasonable that perhaps that pro-inflammatory state might worsen outcome in other diseases.
Absolutely.
I'm wondering, are other people looking at that, or are you looking at that?
I want to look at everything all at once, but I do talk to people from time to time. So like the New York Blood Bank reached out to me when they learned of the work they were doing. No, there aren't enough people looking at it in other diseases, but every now and then someone wanders through my door. So like, for instance, we were studying multiple sclerosis with a postdoc at New York Presbyterian, and he showed that there's a B cell lineage that's unique to Black patients who have multiple sclerosis, and it maybe explains some of the plasticity in that disease. We work with other people who are looking at maternal health. So the inflammation that may occur in the placental tissue could potentially be driving some of the maternal mortality disparities in Black patients. So yeah, we're trying, but at a higher level, I haven't necessarily gotten the ear, if you will, of the chief of medicine at any particular place that could deploy like a system-wide study of just simply tracking Duffy null. I tried at New York Presbyterian, but it's an expensive blood test. So they only do it when it's necessary. And it's like a manual test. And apparently that didn't fit well with the machine. But I think it is going to be an important aspect to inflammation in general. Any disease has an inflammatory component. And so, yeah, we studied it in COVID, actually. So we did see that Duffy null is associated with a higher rate of inflammation in COVID patients in New York.
Thank you.
But happy to do more. I'm really interested in the kidney story. So, yeah.
Any other questions?
Hi, that was such a wonderful talk. Thank you. So building a international or global cohort seems really amazing, obviously, but seems like it must be really difficult. I'm just wondering if you've had difficulties with either the red tape of dealing with institutions in other countries or like just the legality of things, or if for the most part people are very open across the, across institutions, across countries.
Yes, and yes. Yeah, so it is hard. There are definitely unique policies in every place we go. Some of it is national level, some of it is institutional level. One of the things we did for the breast cancer consortium was to work at the highest level. So the Ministry of Health, for instance, is where we would get our studies approved. And then that would trickle down into the hospitals. And so what I found is everywhere we go, if we are in a collaborative spirit and not one of exploitation, then we get a lot more participation. But that's what I want to do anyway. Like, I don't think it's right to just study people and not involve them. And then usually the first thing we do is give something before we ask for anything. But to answer your question about the red tape, I mean, that's just regulation that I actually appreciate because it's a way of protecting populations from being exploited and misused, which is why the IRB is in place now. It can be a challenge, but I think that it's necessary. And I have a team of people who are already doing it and have done it. They have relationships like, Dr. Yates co-leads a prostate consortium across Nigeria and several other African countries. I don't know what the number is now, 7 or something. And working with their relationships and bringing everybody to the table, I think is the way that we've been successful and the way that we'll continue to be. Now, there will always be new rules. Like right now, I thought the biggest challenge would be our African nations, but turns out the UK doesn't want to share any of their data now. So we'll see. We'll see how it goes. But maybe in 5 years, things change. It's always an ever-evolving landscape.
Thank you.
Yep.
All right, one last question.
Sorry, maybe this is a bit opportunistic question, but based on such huge amount of data from so many different planes, do you think it's possible to incorporate it into the machine language model, which will ultimately provide you the tool to provide this targeted, personalized medicine, like all across the so many factors that you're looking at?
Absolutely. Is that what you do?
I wish not.
Yes, but I think it is very important. So one of the things we're going to do, hopefully, I'm waiting to hear back, but AI models that lack diversity at the initial training are the reason why we have the inaccuracies later. Like the AUC curves completely crash as soon as you move outside of the population that it was trained on. One of my favorite papers that I accepted in the journal that I am an associate editor for not only showed that the AI modeling, this was for imaging software across pathology, right? So I think that's where I've seen the like emerging application of AI is through imaging algorithms. But not only did they show that the algorithm was inaccurate in diagnosing African American patients, I think a lot of people have shown that, which is harm, right? I mean, ultimately, if these things are deployed as a standard in the process of diagnostics, we can't ignore the fact that it's misdiagnosing a whole set of people who share something in common. But one of the things they showed was the area of interest that helped to discern the differences between those populations was an immunological response. And so something as simple as making sure that you have people who have that particular immunological response in your training set from a diverse background, because unfortunately, this immunological response seems to be unique to this population. Maybe it's because of Duffy-Null. As long as you have that diversity in the background, I think then, we'll have better tools. So we absolutely hope to feed the machine with more diverse information, but more importantly, inspire others to follow our footsteps in terms of the way we're doing the study. Like, you can't just say it's a hard to reach population and shrug your shoulders and walk away and say, oh, it's too bad, it doesn't work for them. You have to actually dig deeper and do the work, or you shouldn't be allowed to deploy it as standard of care. It's specialty care, as far as I'm concerned. But we do see the FDA like at least attempting to make some adjustments. Some feel it's too little or too late in the phasing, of clinical trials. But we have to start somewhere. I applaud them for even, bringing it up. So yeah, the data will eventually be public-ish. And I hope that it'll seed a couple of generations behind me, to do some great work.
So let's thank Melissa one more time for an amazing lecture.
Thank you for your attention. Thanks for coming. And now the best part is
I'd like to invite everyone to a reception just outside here in the gallery. Lots of food and drink. Please come and partake. Thank you so much.